Okay,
Go ahead.
please, Dr.
Hey.
Bristow.
Thank you, Oswaldo. And so, yeah, welcome everybody. And so, welcome to the RV Falkor 2. We're here off the coast of northern Chile, just off Iquique, for those of you who are locals and know your Chilean cities. I'm Laura Bristow, and I work at the University of Gothenburg in Sweden. But yeah, I'm very British, despite accent, and I am very happy to be here today with Oswaldo.
Okay, well, hello everybody. I'm Oswaldo Ulloa. I work at the University of Concepción in Chile, and well, it is a pleasure to be here on the Falkor 2, sponsored by the Schmidt Ocean Institute, and we want to take you on a tour. Let me just say a few words in Spanish, Laura,
Sure.
and then you correct me if I'm wrong, okay?
Oh, yeah. Definitely.
So, buenas tardes a todos. Aquí estamos con la doctora Laura Bristow. Ella viene de la Universidad de Gothenburg, en Suecia. Estamos en el buque de investigación Falkor 2 del Schmidt Ocean Institute, y lo que vamos a hacer es mostrarles un poco las facilidades que tiene el buque a bordo, algunas de las instrumentaciones, y contarles un poco de lo que estamos haciendo aquí frente a Iquique, en el norte de Chile. Okay, Laura, let's go. You take the lead.
No, yeah. And so, I guess maybe for those of you out there, your first question is, so why are we here? And it's a question we ask ourselves. And so, yeah, as I said, we are here off the coast of northern Chile, and this is a very special part of the world, or one of the very special parts of the world, which has an oxygen minimum zone. And so, what do we mean by that? And so, yeah, below us in kind of the intermediate waters, there are actually no oxygen there. And so, obviously, there's plenty of oxygen here in our atmosphere, allowing us to breathe and survive. But yeah, 100 meters below us, there is no oxygen available for our microbial community and our, that charismatic megafauna that we know and love. And so it can't actually live down there. And these low oxygen regions kind of exist for kind of two big reasons. And so it's because they're kind of in regions that mix very, very poorly. And so we kind of don't see oxygen from the atmosphere or deep waters, new deep waters coming in and replenishing oxygen. So they're already pretty low, but they're also in kind of very productive regions of the world. Like here, this is an upwelling region. It's known really well for its kind of fisheries. And because of that, we see a lot of organic matter produced. And kind of as this rains down, it's kind of decomposed, which means kind of we're using up oxygen even more. And as a result, oxygen, is below the level of detection. And so, what does this mean? And that's kind of one of the big questions that we have. And kind of, we call ourselves microbial oceanographers. And so, these regions are not dead. They are thriving with life. And that's because, there's like millions and millions of microbes there and kind of turning over elements in really different ways than they would when oxygen is available. And that's something that Oswaldo and I and the other scientists on board are kind of really interested in understanding. And so...
Yeah, in fact, we were just discussing with Laura the time we met. That was a long time ago, sometime. It was 2011.
Yes.
And actually, we have been studying oxygen minimum zones for a long time now. We are in love with microbes, and they bring us here. So this is what we do. We try to look at the microcosm, at this very, very small things, but we are very, very excited about what we do, and we think it's important.
Definitely.
Not only because we want to understand how the ocean works, but also because it can give us a hint about how the ocean could have worked in the past, and also with climate change, we know that these low oxygen regions are expanding, so we want to make sure that we understand somehow how they work to make some predictions. Así que bueno, lo que les estaba contando es que con Laura ya nos conocemos hace muchos años, desde el 2011, cuando ella vino a Chile, justamente frente a Iquique, a trabajar en zonas de mínimo oxígeno. Esta es una de las regiones, de las 3 grandes regiones que existen en el mundo, y justamente los investigadores del mundo vienen a trabajar a Chile a estudiar estas zonas de bajo oxígeno, que entre otras cosas nos permiten hacer una mirada al pasado, cuando los océanos carecían de oxígeno, pero también al futuro, ya que debido al cambio climático, estas zonas de mínimo oxígeno se están expandiendo y queremos entender entonces cómo están funcionando. Así que, Laura, let's show them a bit the vessel, this
wonderful vessel for sure. And so definitely the luxurious vessel I've been on.
Me too. So.
And so, yeah, I think it would be great. We should go on a tour and, think about how we actually sample some of these low oxygen waters. And I think we'll also talk along the way about some of the challenges, actually, about how do you kind of get water that has no oxygen in it? How do you bring it up into an oxygenated atmosphere? And it's one of the challenges that we really think about every day out here. And we'll kind of show you some of the tips and tricks and instrumentation that we have to kind of help us do that in the most efficient way.
Yes, indeed. We have here, groundbreaking technology. We're trying new technology, particularly Laura and Maria, which are the chief scientists of this expedition. So we are testing, and as Laura mentioned before, one of the tricks is to measure very, very low oxygen and to get samples without oxygen contamination. So let's start.
No, yeah.
Go ahead.
And so, Oswaldo, I think you should introduce your
No,
baby.
no, you should.
No. And so, Oswaldo, how long have you had this instrument? I remember when we, as you said, when we met, it was with us in 2011, but
yeah,
I guess you had it for a couple of years before that?
Yeah, it has been about 15 years. Okay. At that time, one of the things, the same as today, we wanted to bring water from, let's say, 100 meters with minimum oxygen contamination. So this is a bit of unusual way of sampling. I think there are not too many of these instruments in the world. I think there are three, to my understanding. And what it does is that it pumps water directly from the depth. So you put the instrument in the water, and it has sensors, but also a pump. Yeah. So you bring it through a tubing directly to the lab. I will show you the lab later. And the idea is that that water that we're bringing doesn't get exposed to atmospheric oxygen. We still get some oxygen there, and Laura will explain you later how now we're doing with the problem. But this is one of the instruments that we brought as a Chilean team, since we have had it for many, many years. Brevemente, este es el equipo con el cual el equipo chileno está contribuyendo a esta expedición. Es un equipo que permite bombear agua directo desde las capas del océano que tienen poco oxígeno, y lo que hacemos es enviar mediante una bomba a través de un tubo, una manguera, agua que viene sin oxígeno, y que lo que queremos es llevarla a bordo y que no se nos contamine, a la cual no tengamos el ingreso de oxígeno, y que después vamos a ir analizando en los distintos laboratorios. Esta es una de las tecnologías no muy nuevas, pero que solamente sabemos que 3 instituciones en el mundo la tienen. Así que, let's walk. Yeah, we show.
Yeah, let's go in the van.
Yep.
Where the action happens.
Yeah.
And so, you can see here, this is where the water will then come in to the container.
Laura, I think it's better to go in.
Sure.
Yeah. Este es un laboratorio que lo llevamos en las distintas expediciones a bordo, así que, here, salude. Come on, go.
Hello. You guys look busy. Oh, it's even Fabian down the back hiding. And so, yes, welcome to the lab container, where I think these guys are working 24/7. They always are in here. And so as you can see, we're under some very lovely red light. And so as not to expose kind of the samples that we get to kind of ambient light, and so makes it a lot easier. And you can see there's lots of fantastic instrumentation in here, and so here, this is where we kind of measure nutrients, and so we've got those nutrients that are key for life, you know, particularly nitrogen compounds, and here we can measure the concentration of them kind of in the different samples, and so at kind of amazing high resolution with that kind of pumping system that Oswaldo very nicely just introduced you to. And then they're filtering lots of water, and so we can look at that microbial community that we were just talking about and identify all the different kind of species and taxa that are there, and really think about kind of the different processes that they could be undertaking in these really low oxygen waters.
Como les decíamos, lo que en esta expedición lo que podemos hacer es bombear agua directo del mar. El agua se trae aquí a este laboratorio móvil y tenemos varios instrumentos para analizar, entre otras cosas, nutrientes, pero también aquí las colegas están trabajando con muestras de los microorganismos que son el objeto de estudio nuestro. Así que este es un laboratorio que se puede llevar a cualquier buque de investigación, y en este caso lo hemos traído al Falkor 2. Pero obviamente vamos a ir a conocer los fabulosos laboratorios que tiene el Falkor. So let's go to see the other fantastic labs that this
Definitely.
ship has.
I think, maybe we take a look at the CTD first.
Too? Sure, definitely.
And then we can head to the labs.
Yep.
We'll leave these lovely people to their work.
Gracias,
Thank you.
Gracias.
Thank you. And so now we'll start to head inside. And so, here we are in the hangar, as we call it on board. And this has a number of fantastic instruments in. And kind of the first one that we kind of been using on a regular basis. And I think we've done, is it nearly 150 kind of profiles of the water column with this instrument. And this is what we know as the CTD rosette. And that stands for conductivity, temperature and depth. And then it also has all these excellent kind of array of sensors on the bottom that kind of tells us about oxygen concentration and chlorophyll, and so kind of, you know, how much phytoplankton, those primary producers that are there, and also light levels, and really kind of anything that you can think of, we will try and plug in and measure a profile of in the water column. And this can go down to kind of thousands and thousands of meters. We've primarily been interested in just the top 1,000 meters while we've been on this trip. And what happens is it goes down through the water column, and then at a depth interest, and so we look at kind of that oxygen profile, and when we say, we definitely want some water from there to see what's going on and how the community looks, we kind of trigger it, we can kind of directly talk to it, and one of those bottles closes and captures kind of a water sample that we can then bring up to the surface. And then it turns into a big sampling party when it comes on board, and kind of we all queue up in a nice orderly fashion and take samples one by one for kind of different things that we are interested in, from kind of nutrients, the microbial community, carbon compounds, and so everything that we can think of.
Yeah, there's real action going on every time we have a CTD rosette on board, and literally sometimes we do dance.
We do. There is a very big stereo over there that you may have spotted, and there is very loud music, particularly at kind of 6 a.m. when we're all trying to wake up.
Yeah, here, you know, on board we work 24/7, so you will see people at 2, 3 in the morning working with the CTD, and then, you know, so we need to stay awake and working, you know, so...
Yep, a combination of music and caffeine.
Exactly. So, y como decía Laura, este es quizá uno de los principales instrumentos de los oceanógrafos, se llama roseta, y también y que lleva un instrumento que es el CTD, que puede por conductividad, temperatura y presión, que nos permite justamente hacer observaciones en la columna de agua y cerrar cada una de las botellas a distintas profundidades donde colectamos agua y obviamente los microorganismos que queremos estudiar. So let's Oh, they have another. You want to show that?
You
Yeah,
can show.
yeah. No, you should mention because
Of course we should.
this is really the one of the most advanced
Exactly.
equipment that there is on this ship, if not the most
Definitely.
advanced
Yes.
in the world.
It's truly impressive.
Yeah. So.
And so yes, we'd like to introduce you to a friend of ours. This is Sebastian, and this is a ROV, and so remotely operated vehicle. And so yeah, it's kind of, as we said, remotely operated, and so it's deployed out into the ocean, and then you can kind of take it down to your depths of interest and start to take samples from it. And it's got all these different sample collection chambers, and so then we can be collecting them, identifying organisms that are of interest. And I'm sure all of you who are watching have seen some of the fantastic videos that are on the YouTube channel here of kind of different species and new organisms that have been discovered by kind of the deployments of this fantastic ROV that we have on board here on the Falkor 2.
Yes, we are very lucky that the Falkor 2 is spending all this year,
Oh yeah,
2024, exploring and studying the west coast of South America, of Peru and Chile, and of course Sebastian has been one of the key players, in this exploration and discoveries.
Yep.
Yeah.
So
So this is good. So
fantastic.
shall we continue, Laura, or you want to say something?
I think the only thing is, unfortunately, I'm going to maybe have to do a lot of this because we also have some fantastic instrumentation that is unfortunately currently in the water.
Go ahead.
Right now. And so, as you can see, there's a wire here. And so, there's one instrument in the water here. And so, we briefly mentioned it when we were outside, when Oswaldo introduced you to his pump profiling system, is we have a real problem about how to sample these low oxygen waters. So this CTD rosette is fantastic, but we're going to take a sample that's got no oxygen in it and put it in a big plastic bottle. And there's lots of oxygen within that plastic, and it slowly diffuses out into our precious water samples. And so kind of one of the big questions we have as scientists on board is then, are we actually collecting representative samples? And so we're really, as a community, moving towards working in situ, and so therefore actually collecting and processing our samples while underwater still. And so we have one instrument here that's currently in the water, which is known as a microbial sampler, and that one is kind of, when it reaches a particular depth, it starts filtering away, and so filtering litres and litres of water onto a filter paper, and that is then preserved in situ. And so everything is done down underwater, and then we recover it and just put it in the freezer to be taken home to be analysed at a later date.
Yeah, you're putting the lab inside,
exactly.
in the water.
In the water, yep. and then we have a second instrument that was off the back of the ship, and that one is kind of taking it another step. And so for us to really start thinking about those microbes that were being collected on those filters, what are they actually doing? And so there we're actually collecting water in kind of a big glass tube, and then kind of adding different substrates, different kind of compounds that we would like to know how are turned over in these low oxygen waters. And it allows us to then take samples at different times. It's all pre-programmed, and so it's busy working right now, so I can be here with you, and hopefully collecting a great set of samples. And so we can then compare it to kind of samples that we collect with this more traditional system that is kind of seen on all research vessels that you would visit, to see if we see any strong differences between more traditional sampling methods versus the in situ ones, which will really kind of help guide us for kind of future expeditions and how we can kind of go forward, and maybe even allow us to start to rethink how these systems are functioning.
Yeah, so we don't only need to know who is there, but
what are they doing?
What are they doing? And that sounds like a easy question, but it's been taking us what, years, decades.
Yep, we're
to
still
really,
working on it.
We're still working on it. Yeah, definitely. So let's continue with the trip. Yeah.
And so once we've got all of these samples from the pump profiler or from the CTD rosette, they all come inside, and we now show you where some of the real action happens. Oh, okay. Oswaldo, I think you need to do this one in here.
Okay. So one of the one of the groups here is studying the microbes that do photosynthesis. So they use light and they produce oxygen. But one of the questions we have, is how actually the lack of oxygen or the low oxygen is affecting them. As I mentioned before, oxygen minimum zone are expanding, they are showing, so there may be impacting the community that is even at the surface. And that's one of the questions we have for this team. So here we have very advanced optical instrument. We have Peter, he's part of the Chilean team coming from the Catholic University in Santiago in Chile, and then we have Elisa from Spain, from Mexico, Denmark, or and Antonieta, which is our artist at sea. So that's one of the unique things in these cruises with the Schmidt Ocean Institute, is that they invite artists to join on board so they can work with the crew and with the scientists and get inspired of what we're doing, and we also can see through their eyes how they see what we do.
Yep.
And you can see there that Peter is running some samples in this quite sophisticated instrument. It's called a flow cytometer, and you can study microbes at the cell level. So it pushes the cells through a flow, and then you impact them with lasers, and so you can study their size and their optical properties one by one. Thousands of cells you can process thousands of cells, and then of course you have these beautiful images, pictures, and videos that Peter and Antonieta have been recording and studying, where we can see things alive, and also through the eyes of an artist, trying to get a new dimension of what's the ocean and who lives there. Mm-hmm. So, aquí estamos, ¿cierto? un saludo para todos los que nos están siguiendo.
Okay.
Shall we continue?
And the next place we'll show you is the cold lab. And so before I open the door, and so this is a temperature controlled room. And so, we've been talking about oxygen, how that changes, but also it's actually colder down there, isn't it? And so as you go deeper and deeper in the ocean, it gets colder and colder. And so while it's about, I don't know, maybe I'm being wishful thinking, it's 20 degrees on the surface here, but down there it's about 12 degrees C. And so we tend to, when we bring up samples, we actually then want to work at kind of that temperature, so we're not kind of causing too many temperature changes in those samples that we're collecting. So we collect them from outside, and then we bring them into here. And so, and so here we are.
I'll stay here.
And here we have some people hard at work. And so this is Alyssa and Sina, and they have been sampling this morning, and they are actually looking at nitrous oxide. And so that's a really important greenhouse gas, and so much more potent than carbon dioxide, which I think we talk a lot about when we kind of talk about climate change. But particularly in these low oxygen regions, we see a huge amount of nitrous oxide being produced. And so we've been spending a lot of time here kind of looking at both production and consumption processes of nitrous oxide. And in perfect timing, action shot, they're both horrified. And so what are they doing? And so as we talked about with some of the sampling that we do, we potentially contaminate our samples with oxygen. And so now we're actually trying to get that oxygen back out again. And so we kind of use helium or nitrogen gas to drive out that oxygen in the samples. And now they're, just trying to equilibrate a headspace in the water sample. And they were telling me the other day that they think on the 4 weeks we've been out here, they've spent about 5 hours, each of them, shaking samples. And so, they've got pretty strong arm muscles, I can tell you that. But this is certainly a very popular place to be seen. We have a number of groups working in here, doing different experiments. And so, it's a fantastic facility to have on board, to have kind of this temperature-controlled room. And so I'm going to let Alex come back out of the cold. Okay. And so.
So we're now slowly moving into the main lab.
Mhm.
That's where a lot of the action takes place. So here we are. So right now it's not full, but usually there are many groups
Mhm.
working here, and you can see there's a lot of equipment, and we do a lot of filtering and fixing of our samples here. So and you see that you have some gases there, and all the material that we normally have in a lab.
Oh yeah.
So this is our main lab,
Mhm.
where we do a lot of our work, as I said, 24/7 here.
Mhm.
And so one of the nice things too on the ship is that we can always see what's happening, because they have TV screens everywhere. So and so that's very nice that we
Definitely.
can communicate. This is really a fantastic vessels, you know.
Oh yeah. It's a
I mean,
fantastic space.
the only reason we can be here, you know, alive with you is because they have a beautiful internet
Oh yeah.
connection. We can really communicate and be in contact with
With the outside world.
the outside world.
Mhm.
Something that it was not like that only
Oh,
a few years ago,
yeah.
or in other vessels.
Yeah.
So
I mean,
we are
this
very
is.
lucky.
I mean, this is the best internet I've ever had. This is it's crazy.
Better than home.
Exactly. It means that we can say Happy Mother's Day to everybody out there.
Yes, yes. Happy Mother's Day too. Yes. Okay. So what
Oh, yeah.
else can you tell us, Laura, about?
And so this is where we are doing a lot of sample processing, and so a lot of the magic is going to happen back at home. And so we need to get the samples into a place that we can get them there. And so whether it's filtering or putting them into different containers and freezing them with all of these fantastic array of freezers behind us. And so and then they will all come back to different parts of the world and allow us to analyze, thinking about the community, element turnover, and for us to really, and it's stay tuned. And so give us maybe 6 months. Maybe that's optimistic.
Yes. But you know what? One of the nicest things about this cruise is that we have scientists from institutions from 5 countries.
Is it 5?
From
Yeah.
institutions.
Okay.
If we just look at nationality,
Okay. Yep.
that's a
It's-
bigger
yeah,
number. And that's, I
definitely.
think, is very nice about the way we do science, and about, you know, we interact in a place, in a cruise like this.
No, definitely. It's great to come together and see new and old faces. And and also get the chance to work with kind of the up and coming scientists as they're doing their like PhDs, and look really forward to seeing how their careers come, and watching their data and their careers blossom in the future.
Yes, in fact, I'm the granddad here in
Yeah,
the
he
cruise.
keeps saying this. I don't know what that makes me.
No, you could be my daughter, Laura. Come on, come on, come on. But you are the chief scientist already, with Maria. So come on, that's fantastic. But it's incredible how we have the young generation here coming
Yep.
coming with us, and so I think the future is quite
Oh, definitely. The
promising.
future is bright.
It's bright,
And
Yeah, so it's and that's how science works. Fortunately, it's international. At least what we do.
No, definitely, and it's really nice, both to work internationally and also interdisciplinary, not just kind of doing exactly. No one's doing exactly what each other are doing, so allowing us to really come together and really get a really broad, in-depth understanding of these systems by kind of bringing our different skills and techniques together.
Yep, and we are also at the frontier with technology, and that's very exciting. That's really at least touches me. Yeah, to see how technology is being developed.
Yep.
For science, trying to answer some of the questions that we have.
Allowing us then to really think about kind of what the future holds, and if we're going to make any predictions for both our ocean and our atmosphere, and and the Earth in general, and think coming out of that.
Yeah, and we need them. Otherwise, we won't get the answers we need.
Yep.
So it's that's the challenge.
Yes.
Yeah. So let's
Yeah.
show
Should
them.
we go and have a look in the computer lab?
Yes.
And
let's
then go upstairs?
The
Does that sound
control room.
Yeah.
Okay, let's go.
Oh yeah. You should see the big array of screens.
Yes. Oops.
Because I think this room is really special.
It is.
Compared to so many on other vessels, these rooms are so small and we get all together because, okay, it's quiet now because there's not a huge amount happening, but when the CTD rosette is in the water, this is where we all kind of sit around, and you can see here from a profile we did earlier today, and so we all sit here and watch these squiggly lines and think about where we would like to sample. And so, yeah, thinking about the oxygen profile, which is this light blue line, and so you can really see how it disappears at around 100 meters depth. And then you can put these pink lines here. This is where we collected those water samples, and so close those big grey plastic bottles we were looking at outside, and bringing those water samples up onto the deck and letting us to collect samples. And yeah, this is a great room to be in when that's happening.
Yeah.
And and you get a full on.
So this is where Laura sits,
I
and
sit
controls everything. Yeah.
Be like. Be like, this is where I want to sample.
Excellent.
And so, and yeah, we have a fantastic group of marine technicians on board, and it wouldn't be possible without them to help us kind of keeping this instrumentation in fantastic conditions, and yeah, running these systems for us. It's certainly not just the scientists on board who are making the magic happen, and so yeah, this wouldn't be possible without them.
Definitely. Yes, and again, and also they have been very generous teaching the new scientists, and the new people how the system works, and help them with understanding all the instrumentation that is on board, all the things that we can measure, and the vessel can measure. So yeah, this is very impressive. I think sometimes we tend to envy the people that go into space with their space control room, but this is quite.
We're getting close.
We're getting close. Yeah, we are exploring too.
No, definitely.
So much unknown about the oceans still that we need to continue exploring, and that's what Falkor 2 is doing. Exploring, going to.
Pushing boundaries.
Yeah, exactly. So lucky to be here.
Definitely.
Privileged.
Okay.
To be here.
Are we going to make the big climb?
Yes, I think we are.
Okay, right then. We're going to head up to the bridge now, and so it's a few decks up, so you're going to have to bear with Oswaldo and me if you hear any, we're trying to be fit, but no. But yeah, we're just kind of heading up, and so through some different decks here. Here's kind of where a lot of the cabins where the scientists sleep are, and this is where we muster when we practice, in case we need to go in the life raft. Hopefully that will never happen in real life. And then on this deck is where you find one of the most fantastic things about this vessel is all the great space they have for us to kind of relax in all that downtime we have.
And the excellent food.
And the excellent food, exactly. And we are very well looked after here. We cannot complain in the slightest. And so, maybe only that the bridge is so high up. One more deck to go. And so here we are. One more to go. And so, this is where a lot of the crew have their cabins. So, one final staircase. And here we are on the bridge. Which is the best view of the house, for sure.
Wow.
So.
You have a 360 degrees view.
Yes. And so they can keep an eye on us at all times. Keep us in line.
Exactly.
So don't cause too much trouble. And so, hello.
Hola. Hola, Bob.
But yes, this is where they oversee all the operations. And keep an eye on us and do the fantastic navigation to get us from one place to another and keep everything running. And making this the true success that it's been. And we couldn't have done it without them.
Yeah, so you here you can see that you can see all the action that takes place.
Oh yeah. And all our bird
And
friends. We have a lot of friends on board at the moment.
And well, we had the visit of whales,
We did.
turtles.
Oh yeah. There was a really big pod of pilot whales earlier. Luckily, just as we were having lunch. It was very convenient. Doing. Okay. And so I think it might be question time. And so,
Yes.
yeah, if anybody has any questions, please write them down and we will do our very, very best to answer them.
Please.
And yeah, this is also very luxurious up here. And so.
I feel like being in a TV set,
No, yeah.
So, what questions do we have?
And so, is there anything completely surprising or unexpected that we have found from sampling?
Definitely. And I think you are the person to answer that, Laura, because I think with your instrumentation, you have made some measurements that are, unique.
And so yeah, and so I think as Oswaldo mentioned, kind of, we're trying to, when we say that oxygen concentrations are low, what do we mean by that? And so we have been relying on kind of standard, commercially available instrumentation to tell us how much oxygen is there. But we've actually just been developing a new sensor which has kind of orders of magnitude lower detection limit and higher resolution, allowing us to really capture a kind of a new picture actually of the oxygen dynamics in this region. And so while we actually have kind of relatively maybe large patches that are still kind of below the detection even of this instrument, but that's kind of what we thought was for hundreds of meters of the water column, but it actually seems we see kind of these injections of oxygen and only very small amounts, but they could be really significant for kind of our microbial community. And I think we have really kind of been really surprised about how many we have seen and how often we've seen these injections of oxygen, how long they've lasted, or some have really kind of come and gone really, really quickly. And I think, yeah, we're really excited to think about the spatial and temporal kind of resolution of these and the impacts that it could have.
When remember, Laura, that when we started working on oxygen minimum zones, we thought that they had low oxygen, but they had some oxygen. At least that's what the scientific community believe.
Yep.
And slowly and slowly we have been showing that in fact there's absolutely no oxygen.
No, yeah, in very large patches.
And now you're showing that that's not even the full picture. That in some occasions we do get injections, and we have to try to understand the mechanism why and the impact, but how they are produced, what are the processes, and what are the consequences of those injections. Another question?
Yeah, I mean, they're impressed with the view, so are we.
Okay.
And so, yeah, we have another question of what ways can you envision for the future to visualize microbe collection for kind of a live viewing audience as well, maybe, yeah.
Well, that's a good question.
That's a good question. If they have any suggestions, we'd love to hear.
Well, I think in some ways, of course, we need to develop new technology, images that we can collect, but also I think that bringing an artist on board could
to visualize
also
it,
to show that invisible world to all of us.
Definitely.
So I think there's room, a lot of room for yet to develop in those terms.
And so then, yeah, we've got some questions about the photosynthetic algae that we were talking about with Peter downstairs.
Okay.
And so asking kind of, given how clear the water is, and so, kind of how do we think that might impact the oxygen levels, and so potentially within this region? Because, I mean, how deep do you think we see them down to?
Well, the thing is that in this region you probably see them down to 60 meters, but what we don't understand is that they disappear before light disappears, and there are nutrients down there.
Yep.
And that's something that we really don't understand. I mean, they have light and they have nutrients. So you would expect to have more of
Yep.
these microalgae, and they are disappearing when oxygen is disappearing. So I mean that's something that we really want to understand, what's happening with them and how oxygen is impacting the phytoplankton.
Hmm. No, definitely. And I think, yeah, someone followed on from our discussion of oxygen intrusions, injections that we were talking about. And I think, yeah, they've kind of commented that, yeah, they think that they probably wouldn't be in the environment for very long. And I think, yeah, that's spot on.
Yeah.
We completely agree with you. And I think it's something we're trying to understand because I think you're right. If there was an injection of oxygen, there would be a huge number of members of the community kind of queuing up, kind of fighting, throw those elbows around. I want that oxygen for myself because it can be a lot more there can be more energy for kind of an organism to utilize oxygen than kind of other compounds that would be available. And so it really would, I think, be a fight. And so I think, yeah, it's going to be interesting for us to think about who are going to be the winners there and be the most kind of competitive to take advantage of that oxygen and what it might mean then for kind of how the system is functioning.
Yeah, and I just want to stress the point that without the new technology, we wouldn't have seen that. Because with the standard oxygen sensors that we have in the community, you won't be able to see them.
Yep.
That.
Hmm.
So we are going into,
We're
very
we're moving in a good
moving.
right direction.
Yeah.
And so, and yeah, now people would know about our microbial community.
Ooh.
And so, exactly, Osvaldo's the perfect person. So, keep these questions coming. And so, are there any interesting trends that you have seen in community composition? And so, particular, like, is there any particular dominance of a phyla kind of that you see in these waters?
Well, that's something that we have been studying for some time now, and definitely you have particular groups of microbes that love
Yep.
to be in places like this one
Yep.
where there's no oxygen, which is understandable because you know the evolution of our planet happened without oxygen in the first half of it.
Hmm.
So so yes, there are some particular groups. Um, the diversity is huge. So
Oh,
we are
yeah.
still discovering new groups. But not only that, what really want to understand is what they do.
Yep. And I think versatility. I
Exactly.
think that's the thing that
Yes.
amazes me, right? Is they never stop surprising me how versatile microbes are. They always find a way.
Exactly. Yeah. And now, I mean, just talking about those intrusions that we have discovered here, uh, are they some of the microbes taking advantage, or are they facultative?
Yep.
They can
Mhm.
take advantage of
Exactly.
those spikes of oxygen, or are they being replaced? We don't know.
Oh, yeah.
So many questions about
Many
that.
questions and exciting cruises to come, I hope, as well. And so, and then, yeah, kind of following on, kind of continue to talk about the microbial community. And so, yeah, someone is asking about kind of the chemoautotrophs and wondering if they are similar to ones that we see at hydrothermal vents, or if there is, so is there any crossover that we see, or are we seeing very kind of distinct communities there?
Well, I think that one of the beauties about science is that we tend to find some general patterns. So, in fact, there are some similarities with hydrothermal vent communities. That's something that some groups have been studying
Yep.
in the past, and they turn to use some of the you know nutrients and energy sources like hydrothermal
Yep.
vents, like I don't know, sulfur, for example.
Hmm.
So there are some similarities, but also there are some groups that are specific to here.
Yep.
And more than that, I think there are processes here that we really don't understand yet, and we want to
Oh,
really
yeah.
try to get deeper into that. And
Definitely.
I think, and as I said, one of the challenges has been to work with them with no oxygen
Yep.
contamination. And so we still, pushing the
Definitely.
frontier
Yep.
to try to understand that. But
Definitely.
yeah, but yes, in summary, there are some similarities of both the organisms and the processes that take place in different oxygen depleted systems,
Yep.
like fjords and oxygen minimum zones and even sediments or lakes.
Oh, hmm.
But there are some special
Special.
processes that
Hmm.
take place here.
Yeah.
That impact, the interesting thing is that I think there's less than 1% of the volume of the ocean
Yep.
is low oxygen water. But the impact is global. The
Yeah.
impact, for example, the nitrogen cycle. You are the
Yep.
expert on the nitrogen cycle at the planetary scale.
Yes.
And that's fascinating. And that's why we need to come and
Oh
study
no,
that.
yeah. And so while it's a relatively small volume, the ecological significance really it's huge. And so yeah, and especially as you were saying about these regions expanding, then it's really important for us to think about kind of the impacts that it could be happening. And I think actually you kind of already answered the next question that kind of came up is kind of are kind of these microbes kind of regionally constrained or kind of more cosmopolitan? And I think maybe we should just kind of say that it's not just that there are low oxygen waters here off of northern Chile, and so they are in other places, like you said, in fjords and coastal zones, but kind of in our open ocean, you find these low oxygen waters. So here, kind of off Chile and Peru, but then you also find some in the North Pacific off the coast of Mexico, and then further afield, so in the Benguela upwelling, that's kind of off Namibia, and then also in the Arabian Sea, and also in the Bay of Bengal, and so kind of on the east and west coast of India. And we are seeing kind of strong similarities kind of from what we've seen from the community in these low oxygen waters, that kind of the same players are kind of popping up. It's not that we're seeing different ones here to ones off of Mexico. We're seeing kind of very common features, which hopefully means kind of, the work that we are doing here, we will think of it then is going to be applicable, and so to all of these low oxygen regions, and not just this kind of one spot in the ocean.
Yep, agree totally. Any question in Spanish?
No, not yet. Otherwise, I would have asked you to put your glasses on and read it.
But yeah. Si hay alguna pregunta en castellano, aquí estamos dispuestos a contestarla, así que sin timidez, pregunten nomás.
And so someone has kind of asked, is it possible to do an experiment down these areas? And so yes, we have a in situ incubator. That's what we call it. It has a nickname called Cocktail. So we love it here when they say cocktail on deck. And that is, it's an underwater laboratory. And so it's kind of, this is one of its first outings. And we are very happy that the Schmidt Ocean Institute gave us the opportunity to come out here and really put it through its paces for one of the very first times. And so it goes down to kind of those low oxygen waters and sits there and does an experiment. And so it kind of pulls in water into a glass chamber, and everything is glass and metal, because we don't want any plastic or rubber, because you can get oxygen diffusing out of it. And so then in these glass chambers we can take samples and then kind of add what we call a tracer, allowing us to kind of understand what processes the microbes are undertaking. And then it kind of takes samples and preserves them in situ, and it's out there right now, which is why unfortunately we haven't been able to show you on this tour. It's in the water right now working away, and it goes in the water for about 30 hours at a time, and then we recover it and take the samples off of it, and then it goes back out and kind of does it all over again. And so we're really looking forward to see what it finds.
Yeah, and two things I want to remind you, Laura. One is that this has been taking years to develop.
It's true. Oswaldo's seen kind of the very early version, and so so that's probably like 15 years we've been talking about doing this, and I think we're finally in a place where we really feel good about it. And definitely, if you're going to be any of you out there who want to be a scientist, perseverance is a key trait.
and the other thing too is that I think, to me at least, this is the first time that I'm in a cruise where we can be at a certain place deploying this type of instrument for so long. Usually, there's a lot of demand for the ship time, but this is, I mean, the opportunity that the Schmidt Ocean Institute has given us to test this technology in the proper way. You need time.
No, definitely.
You need many days,
Yes.
and that has been also very unique about this expedition. That we are here doing what we want to do and have the whole ship for us.
No, it's been a fantastic opportunity, and the support we've received is been outstanding. And so and then we have a question about then what is the limiting nutrient or element, and so to productivity. And so then is it or our microbes, and so thinking about is it oxygen or is it something else, like they've suggested iron, for example?
Well, this is a very productive area anyway.
Yep,
So that,
it is.
we have in an upwelling, coastal upwelling area. So this is one of the most productive areas in the world. So you have could be iron limitation. In fact, there are some evidence that in this region there is iron
Okay, I
limitation.
didn't know that.
Yeah, and that's why the productivity is in a very narrow band by the coast,
Very
because
close
we
to the
have
coast.
we have a small continental shelf, and there are no rivers here, and so there is evidence that in this region we have some iron limitation. So that could be the the limiting nutrient here in this region. Yep, that's correct. Yeah, good question.
No, definitely, yeah. And I think everyone's just very excited that we're having a conversation from the middle of the ocean. Well, so are we.
So yes, yeah, yes. Okay, so
Okay.
let's take the last two questions.
No,
So
yeah. At the moment, I don't think we have any. I think we've covered everything.
okay,
And
that's great
so,
then.
but no. And so,
Okay, well, we need to thank the audience here
no,
for.
definitely. If anyone has any final questions, type quickly and we will do our best to answer it. And if not, we need to gear up for our most favourite time of day, dinner. It becomes very important out here, meal times.
Yes.
And so,
And again, we have been extremely lucky to have a wonderful team in the kitchen.
oh, definitely.
You know,
And so,
feeding us so
especially,
well.
yeah, as Osvaldo said, we are working 24/7 here. And so, yeah, if the food wasn't good and had constant supply, it'd be on the floor. And so, yeah, a great question here. And so, how much does the oxygen zone vary by season, or is it more persistent where we are right now?
Well, it's permanent. There are other regions, for example, of Concepcion, where
Yes.
you and I have been working, where it's seasonal, but here is permanent.
Yep.
But of course, its position and size varies. Even, for example, we know that when El Nino comes, everything gets pushed down, so the low oxygen waters are deeper than non-El Nino years.
Yes.
So there is, of course, seasonal inter-area
Yep.
variability, and interdecadal, and so yeah,
Yep.
variability is there. But again, they're permanent here, and that's
Yes.
why we come, because we know
We
that
know.
they are going to be here.
No,
Well, we
yeah.
come and study them. Yeah.
Exactly. It's always, you get a bit nervous when you go to some of the seasonal ones. It's always the fear of like, is it going to be low oxygen or is it going to be fully oxic? And then change our science questions
Yeah.
very quickly if the conditions aren't as we hoped. And so, but yeah. And then, yeah, follow on quickly. Is it current driven? And so, yeah. And so the currents play a very important role here. And so, kind of, yeah, supply, and potentially, yeah, also the reason we're seeing all those oxygen injections that we were talking about. And so, but yeah, it's the currents that are really driving that upwelling and then that high productivity that allows these regions to kind of form and be so persistent. And so, it's really important. And so, and then we have lots of love really from our audience that they want us to keep up the great work and thinks
Okay.
that what we're doing is very cool. But yeah, I think then we will say a big thank you to everybody out there for joining us today.
Yeah.
It has been a great pleasure, and I hope we gave you a good feel for what life on the RV Falkor 2 is like, and some of the work that we've been undertaking out here. And yeah, we feel very privileged to be here, and happy that we were able to share it with you today.
Yes, and in these days where everything is digital, internet, it's great to be out, in the ocean, in
Exactly,
the
yeah.
field, exploring, doing research. So I hope, that the young generations
Yes.
will come and join us and in this endeavor. So yeah,
Thanks so much. Have a great day, everybody. Bye.
adios.